better than a cure? Eradication or elimination of the disease itself? If we could eradicate germs that cause infectious diseases, there would be no diseases to cure. We can say goodbye to infections, symptoms, treatment, and medications. Scientists have been trying to eradicate diseases like malaria, dengue, yellow fever, and zika fever for decades now. You'll be surprised to know that these diseases aren't exactly caused by mosquitoes. It's caused by viruses or parasites living inside female mosquitoes, and when they bite us, the parasite or virus enters our body and causes disease inside us. So if we can eradicate mosquitoes, we can eradicate the diseases that they cause. For this approach, researchers in Florida genetically modified a species of mosquitoes called Aedes aegypti, which is responsible for spreading dengue, zika fever, and yellow fever. Scientists inserted a lethal gene called TTEV into the mosquito eggs. The lethal gene is very dangerous and doesn't allow the mosquito to live up by messing up other genes in the DNA. But what's the point of growing these eggs if they're just going to die? Seems like a waste, doesn't it? So just like we have an antidote for poison, we use tetracycline or an antibiotic to protect the egg from the lethal gene. Once they become adults, scientists release them into the environment. We will see that when these mosquitoes mate, the lethal gene from the DNA will be passed on to their offspring. But there are no scientists around to give them the antidote. So they are not protected from the lethal gene and will not be able to survive. Let's see how with this process, the population of mosquitoes will gradually decrease and hopefully one day we will live in a world without mosquitoes.
Genetically modified mosquitoes. In theory, their widespread release could lead to the total extinction of certain species of mosquitoes, eliminating the vector for so many deadly diseases. It's an extreme idea, but a future with GM mosquitoes flying freely in our world may not be that far away. This summer, genetically modified Aedes aegypti mosquitoes could be released in the US for the first time, in Florida and in Texas. At the heart of the so-called friendly mosquito is something called a self-limiting gene. It starts with Aedes aegypti mosquito eggs, which are injected with short pieces of DNA that contain the self-limiting gene sequence. Once introduced into the mosquito's genome, it encodes a protein that blocks transcriptional machinery for several other genes that are essential to mosquito development. So without essential genes being expressed, the mosquito larvae die before becoming adults. For the gene technology to be useful, the mosquitoes need to be able to pass the lethal gene down to their offspring. And for this reason, the lethal gene has a corresponding antidote. To keep the mosquito larvae with this lethal gene alive, they can be given tetracycline, a common antibiotic. The lethal TTAV protein cannot block the transcription of the other essential mosquito genes when it's bound to tetracycline. This can therefore keep the GM larvae alive until adulthood, when the gene is no longer lethal. So in the lab, mosquito eggs are injected with the self-limiting gene, and the GM mosquito larvae are then reared in water containing tetracycline. This way, they can develop normally into adult mosquitoes. Then, when adult GM mosquitoes are released and breed with wild, non-GM mosquitoes, their offspring inherit the lethal TTAV gene. And without tetracycline in their environment, they don't survive into adulthood. Over time, and with enough GM mosquitoes released, this technology has the ability to hugely suppress the numbers of mosquitoes in the wild.
I don't know about you, but I can't wait for the time when I wouldn't have to worry about mosquito bites and diseases like dengue and malaria. Before you also start dreaming about a mosquito-free world, let's recap what we explored today. First, we saw how Gary Strahan, CEO and founder at Infrared Cameras Inc. in the USA, installed cameras with infrared sensors in public places to prevent the spread of COVID-19. The sensors can detect if a person has fever by calculating the infrared radiation given off by the person. Then we saw how Melissa Finley, veterinarian, and Brian Carson, senior member of technical staff at Sandia National Labs in the USA, developed a portable diagnostic device that could efficiently detect Bacillus anthracis, a bacteria that causes anthrax. The device was inexpensive, simple, and could self-sterilize after its use. Next, we saw how Jeff Galvin, CEO and founder, and David Possa, Chief Science Officer at American Gene Technologies in the USA, found a potential cure for Aedes by genetically engineering T cells of a patient in a way that they can resist and fight HIV. Finally, we saw how researchers in Florida genetically engineer mosquitoes to carry a self-limiting gene that will kill any offspring they produce in the future. This will greatly reduce the number of mosquitoes in the environment and thus reduce the spread of diseases by them. Just like malaria and HIV, do you think we can cure or eradicate any other diseases using gene editing technology? Or could we develop vaccines against such diseases? Maybe one day you can have a breakthrough and cure a disease, saving millions of lives. I hope you liked this episode. Do rate it 5 stars. If you have any ideas to share or any comments on any of the examples we have shown you or got any inspiration, we'd love to hear from you. You can send us a written note, an audio note, or even a video. You can easily attach it in the feedback section of the module. We can help spread your words or ideas by showcasing it on our YouTube channel. So don't hesitate and start sharing. And now, check out the fun DIY activity. I'll see you next time. Until then, keep exploring.